Welcome back to another exciting edition of Ed Puzzle Notes. When we left off, we were talking about Mr. Gregor Mendel, the father of modern genetics. He learned a lot about genetics just by looking at and studying pea plants. He also used other plants, and he tried with mice, but he really liked pea plants because they have very observable traits, things that are really easy to see. Like some pea plants were tall, some were short. That's easy to see. The color of their flowers, the color of their seeds, the shape of the seeds, they're all real easy to see. These are also traits that have just two alleles. So, for like tall, a plant that's tall, it has the tall dominant allele, and the little plants that are short, they have the recessive. So there's only two alleles. So the plants are either going to be tall or they're going to be short. There was also only two phenotypes. The plants were either going to be tall or they were going to be short. There was no medium-sized plant. It was just tall or short. Now, Mendel, if he was going to cross mice, say a black mouse with a white mouse, since the plants were so easy, it was just a dominant and recessive. So either you got a tall tree or a short plant. Well, with other organisms, it's not quite that easy. But if you were to cross a black mouse, and let's say it's homozygous, dominant, big B, with a white mouse, which is little b, if you do a simple Punnett square, all the boxes are going to be the same, so I'm just going to fill one of them in. Big B, little b, don't have to do those. If they're all big B, little b, and since they have that dominant big B allele, 100% of those little baby mice would be black. Well, it's not always that easy. Sometimes when you cross black mouse with a white mouse, or anything, could be a cow, mouse, cat, who knows. But sometimes black crossed with a white, you get gray. Because if you mix the colors black and white, you get gray. So every now and then you get that. And that's called incomplete dominance, which we're going to go over in a second. Also, in some cattle, if you cross a black cow with a white cow, you get a cow that's both black and white. Can't really read that, but both black and white. And that's going to be something called codominance, which I'm going to go over here in a second. So incomplete dominance and codominance. All right, quick review. If you remember from, a couple weeks ago, same has 4 letters. Same has 4. So, when you look at these terms, hetero, the prefix hetero, has 6 letters. That is not 4. So that equals different. The letters are going to be different. The alleles are going to be different. True breed. The prefix true has 4 letters. 4 equals same. So that means the alleles are going to be the same. They're going to be both dominant or both recessive. Pure also has 4. So that equals the same again. Hybrid. That has 6 letters. Since it's not 4, that means different. Homozygous. The prefix homo has 4 letters. Since it has 4 letters, we know it is the same. So different would be like big F, little f. And if you remember, when we're doing these Punnett squares, the monohybrids with just one trait, we start with a genotype of 2 letters, but we got to separate them. Because remember, you get one from mom, one from dad. Well, when you're a mom or a dad someday, you're going to give one to your kid, one or the other. So, let's say big F, little f, heterozygous, and we're going to cross with a homozygous recessive, little f, fill in the boxes, big F, little, big, little, and little. All right, like I said earlier, single gene traits. Just like it says, these are traits that are determined by a single gene. Now remember, we do have 2 of these genes, like this is a chromosome, that'll be from mom. This is a chromosome, these are homologous chromosomes. And we'll say they're chromosome number 5. So they're both chromosome number 5. They're the fifth largest chromosome. So like we'll just say, hey, there's the gene for dimples right there. Hey, it's also going to be right there on chromosome number 5. The gene for dimples is not found anywhere else on that chromosome or on any other chromosomes. It is a single gene trait. One gene controls whether you have dimples or not, and it also controls the number of digits or fingers you got. It's also a single gene trait. You can see this kid's got 1, 2, 3, 4, 5. This kid's got, was it 5? 1, 2, 3, 4, 5. Yeah, all right. And this guy's got 1, 2, 3, 4, 5, and 6 toes. So that is determined by a single gene, and that's called polydactyly if you have 6 fingers. And as you can see, the 6 fingers gene is actually a dominant. So to have 6 fingers, you can be big F, homozygous dominant, or big F, little f. Either way, you will have 6 fingers and 6 toes. Alright, so here's some single gene traits that are very easily observable. First one is what almost everybody can do, the rolling of the tongue. And these are dominant traits, so if you have one of these alleles, remember we have 2 alleles, we get one from mom, one from dad. Well, if your mom or your dad gives this to you, then you can do that. So let's put a T, big T, you can roll your tongue, or big T, little t. The only way you cannot make a little taco or roll your tongue like that, you'd have to be little t, little t. Another dominant single gene trait are these things right there, the dimple. That's located on chromosome number 5. A single little spot on chromosome number 5 has the instructions or the code to make dimples. And that's dominant, so you can have dimples with big D, or big D, little d. Both the same, you'll both have dimples. The only way you don't have dimples like me, boom, little d, little d. You get one little d from your mom, one little d from your dad. Single gene. So these are not controlled by several genes on several different chromosomes. Here's another one. If your second toe is longer than your big toe, that is much more rare. I can't do that. I can roll my tongue, but I cannot do that. But that's another dominant, dominant single gene trait. And a widow's peak is like a little V that you have on the front of your hairline. Now these are different. These are not single gene or monogenetic traits. These are polygenetic. So these traits are determined when you combine a bunch of genes together, a bunch of alleles. So it's not one gene. For example, skin color, geneticists think that the color of your skin is controlled by somewhere in the order of 312. It might be 318, I forget. But on last count, it's the low 300s. That's how many genes control the color of your skin. And that makes sense because pretty much everybody's skin color is a little bit different. Some's a little darker, some's really dark, some's really, really light. Well, all those different variations, those different shades, is because it's polygenetic. Same thing with height. That's why not everybody is 7 foot tall or 3 foot tall. There's so many different variations, 5'6", 5'5", 5'4", 6'3". That's because it's polygenetic. It's controlled by several different genes. Same with eye color. That's why even if you have green eyes, they could be like a darker kind of brownish green, a hazel, super light green. That's because it's controlled by more than one gene. Hair color as well. So these are polygenetic. So the other ones like dimples, that's single gene traits or monogenetic because it's controlled by one gene on one chromosome, where these are controlled by several genes on several different chromosomes. Alright, so now codominance. This is different than simple dominant and recessive because in codominance there is no recessive. Both of the alleles are dominant. For example, pure red, first let's go the old school way. Let's say we crossed a red cow, pure red cow, we'll say that it's recessive, where it's white is recessive, so we'll go little r's. Now this is not how it works with codominance. This is how it would work with simple dominant and recessive. They'd all be big R, little r would equal 100% of the baby cows would be red, because red would be dominant. But in codominance, since both of the alleles are dominant, you don't get two different phenotypes like red and white, but you also get a third phenotype in which both of the parental traits appear together in spots or patches. Now, one way I remember this, instead of thinking of codominance, thinking of it as cow dominance. Cow dominance. Because you'll see on the next page, I'll show you how it works. And speaking of cow, there it is. Codominance. Codominance, or you can think cow, because we've all seen cows look like that. How do you get cows like that? Well, remember, both red and white are both dominant, so there's no more little letters. So doing a little Punnett square, we'll do the dad cow is totally red, he's all red. The mom cow is totally white. Well, when those little baby cows, they're all going to be RW, RW, RW. All the baby cows are going to look like that. So they're all going to be patches, some patches of red, some patches of white. Now, if I cross two of these guys, big R, big W, now, so an RW, red and white cow, crossed with another red and white cow. Now, this 1 out of 4 times, you'll have baby cow that's totally red. 2 out of 4 times, you'll have the red and white cow, like this guy. And 1 out of 4 times, you'll have the all-white cow. So that is codominance, where you have patches. You have patches or areas that have both dominant traits, but they do not blend together. So you don't get pink cows, you get red and white. All right, moving on. Incomplete dominance. It's kind of similar to codominance, but unlike the cows that you saw, codominant, cow dominant, they're red and white. This, in incomplete dominance, this is where you cross, like, say, a red flower with a white flower, and now we don't get patches of red and white. Now we get the pink, where the blending, the traits are blended together, resulting in a third phenotype. So we have 3 phenotypes. We got red, white, and then also pink in this incomplete dominant. So a simple little Punnett square for this one. If you cross a purebred red with a purebred white, just like in the previous example, we get RW in all of them. I don't need to do that one again. So 100 of the flowers will have the genotype of RW, and 100% of them will be pink. That phenotype of being pink. And there it is, purebred red crossed with a purebred white flower. All of their offspring baby flowers will be pink, and that once again is incomplete dominance. Remember, with the cows, cow dominance, that was called codominance. All right, this slide I don't need, so I'm just going to use it to ask you some questions. All right, last concept for today. So far we've seen single gene traits which are determined by a single gene on a single chromosome. Polygenetic traits that are determined by several genes on several different chromosomes. We had incomplete dominance. That was where if you crossed a red flower with a white flower, you get pink flowers. And we also saw codominance. That's where if you cross a red cow with a white cow, you'll get a baby cow that has patches of red and white. That's codominance. Now we're going to look at multiple allelic. Now remember, most of the time you only have 2 different alleles for a trait, like say big D or little d. That's 2. You know, there's a big D, there's a little d. That's 2 different alleles. Or like with red and white, you have R or W. 2 different alleles. Here we're going to have 3 or more alleles to determine this trait. And the only trait we're going to look at as far as multiple allelic is blood type. So most people have heard of the blood types. You can be type A. Well, there's an allele for A, there's an allele for B, and there's another allele for O. Now you could also be AB, type AB blood. That just means you have one type A allele and you have a type B allele. And once again, you get these from your parents. You get one from mom, one from dad. And multiple allelic is also mostly like in fur color in animals, cats, dogs. Those are also multiple allelic. But all you're going to need to know is the blood type. All right, so when it comes to blood type, it is codominant because A and B are both dominant. Let's put dom. O is recessive. That means the only way you can have type O blood is you got to have 2 alleles for type O. Now, people that are type AB, you're kind of cool, even though it's the rarest of blood types. You're what's called a universal acceptor, because you can get any type of blood if you're type AB. You can get blood that's type A, and you'll be fine because you have the allele for type A. You can get type B blood inside of you because you're fine because you have the B allele. You obviously can get type AB blood if you need a transfusion because you're type AB, but you can also get O because O is recessive, and basically anybody can get O. I'm type O. It is the most common. You're called a universal donor because anybody can use your blood. So they love type O because they can just use it. Don't even need to know your blood type. But if you're type O, you can only get type O blood. So you cannot get B, you can't get A, or you can't get AB. You get very sick. You need to get type O if you're type O blood. Now, just really fast, I don't know if you guys remember, but we talked about the jobs of proteins, and one of the jobs of proteins was that they served as like a name tag or an ID card for your cells. Well, your blood cells have little name tags on them. And one of them is the type A name tag. Those are called antigens. And you have the type B antigen. Well, if you have type O blood and you have blood cells cruising around with a type A ID tag, your immune system is going to say, hey, that's the wrong name tag. We don't have type A blood, and your immune system would fight those blood cells, and that's why you would get really sick. So just remember that antigens, those are like the protein little name tags that are on all your cells, including your blood cells. All right, the most difficult slide of the day by far. Remember, our blood type is determined from our parents, what allele we get from our parents. So this is the allele from mom over here, and this is the possible alleles from the father. If the mom gives the allele for type A blood, and the father gives the allele for type A blood, this is the genotype of the kid. Now, you don't need to know what that big I stands for. Just know that the big I means it's dominant. It stands for isoagglutinogen, but you don't need to know that. But you can see that they both have the superscript A's there. That's homozygous for type A. Homozygous, because they're the same. They got 2 type A alleles. Now, the opposite of that, this person got a type B allele from both parents. So that would be their genotype. Big I, big B, big I, big B. Once again, that's homozygous for type B. Homozygous, true breed or pure breed. Now, this person got a little different. He or she got a type A allele from mom, but got a type O allele from dad. This person is not type AO. There's no such thing. So they got the A allele from mom, the recessive O allele from dad. This is also type A, just like that person. They're both type A blood, but this person is heterozygous or hybrid for type A. This person got the B allele from mom, the A allele from dad, so that person would obviously have the blood type of AB. And this is the same thing, just in reverse. Got the A from mom, got the B from dad. Once again, there's the genotype, big I, big A, big I, big B. And there's the phenotype, type AB. This is very similar to the third one. Person gets type B from mom, type O from dad. Here's its genotype. Big I, big B. Got that from mom. Recessive little I, the O allele from dad. Now remember, that's not type BO. That is type B. Heterozygous for type B blood. And the last one, the only way you can get type O blood, you gotta get type O from mom, type O from dad. This would be your genotype, little I, and that is the only way you can get type O blood. All right, you guys probably thought we were done with Punnett squares. Unfortunately, we are not. Now, if you like doing them, if you think they're easy, boom, free points. I'm handing out free points for you. If you don't know how to do them still, no big deal. They're easy. You can still learn them. All you got to do is pay attention and try them. You will learn them. All right, so we're going to do some blood types. We're going to cross, let's say, heterozygous type A. Heterozygous type A. So that's going to be big I, A, and a little I. And we're going to cross that person with a, let's go heterozygous type B. So this could be like the dad right here. Big I, B, little I. All right, and just like a regular Punnett square, we put their alleles on the top and the sides. We're going to go big I, A. That's one allele that can be given to the kid. And little I. And we'll put the moms or dads, whoever this is, on the side. Big I, B, and little I. Then you just fill it in. This kid up here, big I, A, big I, B. That person's going to have type AB. Big I, B, little I. That kid would have heterozygous for type B blood. This kid, big I, A, little I, heterozygous type A blood. And 1 out of 4 chance your kid will have type O. So we got all 3 different phenotypes here. We got type AB. Half or 1/4 of their kids would have type AB. 1/4 of their kids would have type B. 1/4 of their kids would have type A blood. And 1/4 would have type O. Let's try one more. How about type O crossed with a type AB? So the genotype for type O blood, type O blood is little I, little I. And we're going to cross that with a person that is type AB. Big I, big A, big I, big B. All right. Regular Punnett square just like always. Boom, boom. We'll say this is the mom. We'll put her alleles up here. Big I, or little I, little I. Remember, I just want one letter. For monohybrids, I just need one letter above each column and beside each column. Let's put the big I, A. That's one allele. And we got the big I, B. I guess those are 2 letters, but that is a single allele. Now just fill in the box. Big I, A, little I. Big I, A, little I. 50% of their kids would be type A blood, heterozygous for type A. The other 50% would be big I, B, little I, big I, B, little I. So the other 50% of their kids would be heterozygous for type B blood. Most common mistakes, people forget to put the big I. Another thing, people forget that the little I, that is how you signify, that's the allele for type O. So a lot of people give me big I, O. Well, don't give me that, because that's not the allele for type O blood. You got to show that it's recessive by putting a little or lowercase I. So little I, that is the allele for type O blood. Thank you for watching, and I will see you next time. I know this is a lot of stuff, but it's not that difficult.